Welcome students to our beautiful channel Biophilia, the love for biology. Today we will easily study DNA in a very easy way so that it will be helpful for you to memorize. So let's start. This is our cell which contains a nucleus. If we expand the nucleus and see, then we will come to see that it contains various different types of chromosomes. These chromosomes are made up of DNA molecules. DNA is a basic unit of heredity and it passes from one generation to the other, which helps in the carriage of genes from one generation to the others. DNA is made up of majorly three things. A DNA is made up of pentose sugar, phosphoric acid, and nitrogenous bases. Nitrogenous base, that so DNA is composed of three things: pentose sugar, phosphoric acid, and nitrogenous bases. Nitrogenous bases have purines and pyrimidines. Nitrogenous bases have purines and pyrimidines. In purine group, we have adenine and guanine, whereas in pyrimidine group, we have cytosine, thymine, and uracil. DNA is majorly made up of adenine, guanine, cytosine, and thymine new bases, whereas RNA thymine is replaced by uracil. This is a basic component that helps in production of DNA. Now we will study the Nobel Prize and structure of Watson and Crick DNA. Watson and Crick proposed the DNA model in 1957 and got Nobel Prize for this topic. This is our DNA. It is made up of, this is pentose sugar called the ribose sugar. This is the first prime carbon, second prime, third prime, fourth prime, and CH2OH. This is the fifth pentose sugar containing 5 carbon. In the first prime position, we add the nitrogenous base containing adenine, guanine, cytosine, and thymine in case of DNA. In the second prime position, ribose sugar usually has OH, but in case of DNA molecule, the O is removed and only H is present. So thereby it got its name, that is deoxyribo. D means cut and oxy means oxygen. Therefore, as 2' position has no oxygen, therefore we can say that DNA is a deoxyribose sugar. Okay, then we can memorize and the things by the DNA is made up of pentose sugar, nucleotide bases, and phosphoric acid. Nucleotide bases contains purine and pyrimidine, where A and G comes in purine and C, T, and U comes in pyrimidine. This is the basic structure of the ribose sugar. Now we will study the bonds present in DNA. DNA majorly contains three types of bonds, that is phosphodiester bond, glycosidic linkage bond, and hydrogen bonds in them. So this is our ribose sugar. In the 5' position, we have CH2O, P, O, P, O, P, O group. OP, 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 and O group. Double bond O, double bond O, double bond O and single OH molecules. With the first prime, there are nitrogenous bases. Similarly, in this molecule, we also have this type of long chain made up of phosphoric acid. The 3' OH of the first nucleotide molecule reacts with the oxygen group, oxygen of the second molecule, and forms a phosphodiester bond. This bond looks like this. This is our ribose sugar. OPO double bond O double bond O. This is a phosphodiester bond between the 3' and 5' position of the nucleotides. Here we have adenine, let's suppose guanine. This bond is called glycosidic bond. Therefore, we can summarize as the DNA have 3' to 5' phosphodiester bond, and it has glycosidic bond with which nucleotide bases are linked to the first prime carbon. Okay. Now DNA are antiparallel in nature. Here is our adenine molecule. Adenine forms two hydrogen bonds with thymine. Adenine forms two hydrogen bonds with thymine, whereas guanine forms three hydrogen bonds with cytosine. These are the hydrogen bonds represented by the lines. Here we have the phosphodiester bonds. We can see that in this nucleotide we get a free 5' end, whereas here we get a free 3' end. If we see the other strand, then here the 3' end is free, and in this case the 5' end is free. Therefore, we can clearly distinguish that DNA has opposite polarity, as it is here 5' and there it is 3'. Therefore, the DNA is antiparallel in nature. Now, we will study the molecular structure of the bDNA model by Watson and Crick. DNA, there are many types of DNA, namely aDNA, B, C, D and Z DNA. aDNA contains 11 base pair per turns. bDNA contains 10 base pair per turns. C contains 9, D contains 8 and Z has the maximum, that is 12 BP base pair per turns. 12 base pair per turns. This is the structure of DNA. This is the B model. It contains 3, 4, 5, 6, 7, 8, 9 and 10. 10 base pairs per turn. In this turn, it contains 10 base pairs. Scientists have calculated that this length is approximately 34 angstrom. Therefore, if we need to calculate the width of the between these two nucleotide pairs, then it is 3.4 angstroms. This is the molecular structure of DNA with the length and the breadth of 3.4 angstroms. Therefore, we will summarize now. The DNA is made up of majorly three things: pentose sugar, phosphoric acid, and nitrogenous bases. Nitrogenous bases are composed of adenine, guanine, cytosine, and thymine. Whereas adenine and guanine are paired under purine, and cytosine, thymine, uracil are paired under the pyrimidids. RNA contains uracil in place of thymine. DNA is opposite polarity, and now we will Thank you students for joining us in this session. Keep learning and keep growing.